Let me get started. It's my great honor and great pleasure to have Dr. Nancy Zhang joining us today to talk about great stories that have been publishing in really high impact journals over the past many years. So Dr. Zhang obtained her PhD in statistics, doing a lot of bioinformatics in 2005 from Stanford. And then she decided that she needs a postdoc, but for very, very, very short postdoc of 1 year, which is kind of unheard of. So she went to UC Berkeley and then went back to Stanford as an assistant professor in 2006, I believe. She moved up all the ranks, then she moved to UPenn, I assume in 2012 as a tenured professor, and since then have been receiving a lot of awards, just to name few, Sloan Fellowship Award, recently Medallion Lecturer, which is a great in mathematical statistics, and has been involved in the community. And I'm very, very pleased to have Nancy to tell us today about her recent works. And let's welcome Nancy, and floor is yours.
Well, thank you. Thank you so much, Majid, for inviting me, and also to Daisuke and you for hosting this seminar. I would love to visit you guys in person, but I'm grateful for this chance to at least, do this through Zoom. Yeah, so actually my 1 year postdoc was atypical in statistics at that time, because a lot of statistics PhDs graduate and just go directly to faculty, but I needed 1 year to sort of get my act together before starting. So I am giving you a talk on this. This is slightly different from my abstract, and my abstract is on allele-specific copy number analysis integration with chromatin accessibility. I decided to put more recent work into this talk. So it's single cell multimodal DNA copy number analysis. I understand I have 45 minutes. I think my stuff is probably can be finished in 30 to 40 minutes. So, if you have questions, it's always hard in Zoom to know how whether I'm making sense to you. So I would appreciate you, if you have questions, I have the chat window open, so you can just put it in chat or just call out. I won't be offended. So, okay, let's get started. We work with a lot of cancer data. And the first thing to say when we talk about cancer is that it is a very cool evolutionary system, right? This is the intra-tissue evolution, where you have an initial cell that gains some mutations that give it better fitness, make it able to grow and divide and not die, right? And that spawns a primary tumor. And the primary tumor, because of the fast mutation rate in cancers, that's actually a hallmark of cancer, that cancer cells mutate fast. That's how they're able to gain the diversity in the primary tumor, that say if there is you treat it by chemotherapy, oftentimes you don't treat you don't actually kill all the cells, and there will be some rare subpopulation of cells that's able to break free and then see the metastasis. And that's what makes this disease so difficult and so scary, right? So a feature about any tumor is whether it is genomically unstable, meaning that whether it mutates fast. And it turns out that how fast it mutates, we can measure that by the subclonal diversity in the primary tumor, and that is directly linked to clinical outcome. Okay, so there's been many studies about this. Here I'm just showing a figure from one such study 5 years ago, where they actually looked at clonal diversity using bulk sequencing. And you can imagine that's very hard, right? Because you just sequence something in bulk, that gives you the mean profile across the cells in the tumor. And they were able to actually figure figure out for each tumor whether there's more than 2 clones or 2 or less clones. And that that's a very noisy estimate, but it turns out to be enough to see that the tumors with more clones tend to have a worse survival time. This red curve here. Okay, so and then there's other studies like this that give plenty of evidence that the diversity of a tumor, the genetic diversity matters. So, but these are cells that are growing and dividing in tissue, right? So other than DNA level mutations, epigenetic level changes also get passed down through mitosis. So by epigenetics, I mean like methylation and also chromatin accessibility. So the tumor evolution is unique in this way that both DNA level and epigenetic level mutation sort of changes, they intertwine and they together shape the developmental progression of the tumor. So by in this talk, I'm going to focus on a specific type of genetic mutation, which is copy number alteration. And then I'm going to focus on a specific type of epigenetic change, which is chromatin accessibility that we measure using ATAC-seq. And in terms of cancer and the phenotype of the tumor cells, we're going to look at RNA-seq. So we're going to look at tumor cells from 3 angles: the DNA level copy number mutations, the chromatin level accessibility as measured by ATAC, and the RNA sequencing. Okay, so that's sort of the background, a quick background, and here is just a more specific outline of what I'm going to go through. I'm going to start with single-cell DNA-seq data because we are going to be looking at copy number first. And copy number is at the DNA level. And specifically, I'm going to look at allele-specific copy number analysis. And I will tell you why that specific type of copy number analysis is important. And then we're going to apply our tool to ATAC-seq data. I'll show you how it applies. And the bullets 1 and 2 is already published in our recent paper, integrative single-cell analysis of allele-specific copy number alterations and chromatin accessibility in cancer. And so I'm going to provide some examples from that paper, but the details you can look. And then after that, I'm going to give you a rundown of some recent work on subclonal identification and copy number profiling in single-cell RNA-seq. Now, single-cell RNA-seq, as compared to single-cell DNA ATAC-seq, is actually the most mature protocol. It's the one where most people are doing this. It's the easiest to do, actually, as compared to ATAC and DNA, because the protocol is so well developed. But we've been slow in using RNA-seq in our evolutionary analysis because it's much harder, and I'll tell you why it's harder. But recently, we were able to make some progress in that. And the reason why we want to move in that direction is because, if you do single cell or molecular profiling, that everybody is very excited nowadays about spatial transcriptomics, which is where you measure high resolution, almost single cell level, the transcriptomes of cells fixed in space. So the spatial coordinates. And you can imagine how important that is for subclonal and evolutionary analysis of cancers, because things sort of spread across space. So we want to apply our ideas and concepts, that's developed for single-cell RNA-seq to spatial transcriptomic data. And this is all very fresh and sort of unpolished work from our lab. So I'll just be giving you a brief overview. Okay, single-cell DNA-seq, right? So the first paper that was able to isolate single nuclei from tumors and then sequence them was in 2011. And this is a paper by Nick Navins, when he was a postdoc. So they sequenced, I think, like on the order of 100 nuclei from a breast cancer. Okay, and then they estimated the DNA copy number profile of these nuclei. So since then, it's been 10 years, but methodologically, compared to the number of methods that's been developed for single-cell RNA-seq, there hasn't really been as much activity in this domain. And that's because the technology, the technology development has been kind of slow. Recently, there's still papers that's been submitted to science journals about a new protocol for doing single-cell DNA-seq. There's been quite a number of methods for DNA copy number analysis using hidden Markov models or window smoothing approaches. So my method, we call it Alleloscope. Okay, and Alleloscope, basically the innovation here is that we try to go for allele-specific copy number operations. And also we developed this method to be able to apply to low coverage single cell sequencing. Okay, low coverage meaning for each cell you don't get much reads, and that makes it applicable to also to single cell Ataxi data. Okay, so let me first tell you about allele-specific signals. So what does that mean, right? So each cell in our body, right, is under the normal circumstance where there's no disease, should have two haplotypes, right? One from the mother and one from the father. And we denote at heterozygous sites, we denote the two alleles by A and B, right? And the B allele frequency is the proportion of alleles that is B, right? So at this SNP, it should be 1/2, right? Because there's one of each. And then at each SNP, if it's diploid, it should be 1/2 the B allele frequency. But then when there is amplification, right? If the blue haplotype is amplified, then the B allele frequency for this specific SNP will be 1/3, right? And then at this SNP, it will be 2/3. So it depends on which haplotype the B allele lands on, right? And then if there's a deletion, then the B allele frequency goes from 1/2 to either 0 or 1, 0 if the B allele is deleted, 1 if the A allele is deleted. Now, one of the main reasons, in the early years when we started looking at allele signals is because oftentimes in cancer, you can have what's called a copy neutral LH event, which is shown here, where you lose the blue copy, but you gain the red copy to compensate. And so the total copy number is still 2, but you have lost the heterozygosity. So you have 2 copies of the B allele or 0 copies of the B allele. So why is that? What does that matter, right? You still have 2 copies. Well, you can imagine that, this is often the case, it's kind of scary to think about that. Each of our genomes actually carry a lot of loss of function mutations. And, all of us carry these loss of function mutations. But the reason why we're still healthy is because we have another normal copy from the other parent, and that makes up for it. But you can imagine that if we lose that normal copy, and then even though we have 2 copies of the chromosome, both copies have this deleterious mutation, and then that leads to disease, right? So that's oftentimes the route by which we see this in real data in empirical examples that cancer cells lose their normal function through this mechanism. And another thing that happens in cancers is that you could have a gain of function mutation in one of the haplotypes that makes the cell produce some version of the gene that makes it more fit, right? So a gain of function mutation that makes the cell divide faster or whatever. And then when the cancer progresses, this gain of function mutation actually gets amplified to many copies to even further improve the fitness of the cell. So you see that alleles, knowing which allele gets affected does matter in a cancer setting. And here's just another reason why we care about allele-specific copy number. So we knew this in the days of bulk sequencing. So here I'm just showing you a figure from a bulk sequencing study in 2017, where they took lung cancer and they sequenced multiple regions. So here they took 4 regions of this cancer. And because they knew that there was intra-tumor heterogeneity, that not all cells are the same. And at that time, the single cell, even though, the single cell technology is already out, not every lab knew how to do it. So they did bulk multi-region sequencing, and they conducted bulk allele-specific analysis. And what they found is that there's this region where there's an amplification. So, in normal germline, the BAF is at 0.5 as it should be. It's an amplification, but between the two regions of the cancer, a different haplotype is amplified. So in one region, the maternal haplotype is amplified. So the orange has a 2/3, and then the other region, the paternal allele is amplified. So it's blue that is on top. And that's pretty surprising because, if you were to look at total copy number, you don't see that there's actually two different subclones, that genetically they're actually different. So you wouldn't be able to see this even if you were looking at single cell data, if you were only computing total copy number, right? Because if you're only computing total copy number, these two will still be the same, even at a single cell level. So that's why you have to be looking at single cell or multi-region sequencing and looking at allele-specific changes. So before our study, there were a few studies that sort of highlighted these mirrored subclones, but they went undetected and really understudied because they're just, it's so hard to study them. So I want to mention this study, which is a very nice paper published February of this year. They were doing something that was very similar to what we were trying to do, which is allele and haplotype specific copy numbers in single cells. And their method is called CHISEL. And what they found, they looked at one very high coverage breast cancer sample at a single cell level, and they were able to find 2 instances of such mirrored subclones in the breast cancer. So that just, tells you that these events do exist. But, we felt that there was still a big gap in terms of methodology in doing this because this CHISEL method, they were trying to look at allele specific copy number, but they could only be applicable to, because of the way that their model works, they were only applicable to very high coverage single cell DNA sequencing data. So which is, what they did in their study. But most data that is out there and that labs can afford to do is either very low coverage single cell DNA sequencing data or ATAC-seq or other not single DNA. And these data don't have the reads that allow this method to work well. And further, they rely on external phasing at heterozygous sites. So using HapMap phasing to help you connect the alleles at different SNPs. And that we don't always want to rely on external phasing because the sample you're looking at may be from a population that is not very well covered by HapMap. So because of these reasons, we worked harder at our method to try to address these issues. So before jumping into our method, I want to give you sort of a cartoon overview of what this problem entails. So what is the underlying structure of single cell allele specific data? Okay, so let's imagine that among your cells, you have the normal subpopulation that is basically deployed one copy of each haplotype and everything, there's no copy number changes. And then let's say that there is another subclone of tumor subclone that contains, just say, a LOH of this right region here, where instead of a blue and a red, you now have 2 red haplotypes. And let's say there's so it's right here highlighted. And then let's say there's another subclone that doesn't have this LOH event, but it contains an amplification of the red haplotype that overlaps with it. Now, this is all just hypothetical. I'm just showing you an example. And in the real data, you would never know about these because they're latent structure in the data that you want to recover. So what do you actually observe in the real data? In the real data, in the observed data, you don't know the breakpoints. You don't know the haplotype, the phasing, and you don't know how many such subpopulations there are. What you observe are reads from single cells. So let's say for cell 1, you observe these reads that are shown as tick marks. And if the read does not fall on top of a heterozygous site, then it registers a count, but you don't know which allele it contains. But if a read overlaps with a heterozygous site, then you can say this has a B or the A allele. And then you have such data across many cells, and across cells, the reads don't line up because the reads are just, you just fragment the genome and, they land where they land. And so you may have reads at different locations in different cells. And the nice thing about single cell data is that you have many such cells. So in a typical study, in a single cell DNA study, you have hundreds of such cells. In a single cell Ataxi-seq data, you can have thousands of such cells, right? So you try to pull information across cells to detect the underlying clones, but for each cell, it's very sparsely sequenced. So what is hard about this data? Well, there's, the challenges are many. One challenge is that within each cell, the reads are very sparse, okay? And the second thing is that you don't know where the breakpoints are, right? So you don't know a priori, at least for single, without, say this region is an LH and this region is an amplification. And furthermore, we don't know the underlying phase. Now that is a big problem because the data is so sparse. The way that most methods for total copy number analysis work is that you sort of pull across adjacent regions, right? The copy number changes are fairly large, so you try to make use of count reads across a bin or something. But because we don't know whether, say, the A allele for this SNP is on the string together with the A allele of this SNP, and so that the haplotype is AABB, so you're supposed to string together those SNPs, those alleles, but we don't know that, right? So it could be that the A and the B are aligned in a haplotype. So that's what I mean by phase. And if you don't know the phase, it's hard to know how to combine across different alleles across adjacent SNPs. So that we built a model to try to address this problem. And I know that there's quite a number of mathematicians and computer scientists in the audience. I'm going to go through the model really quickly. So our model is that for each cell I, the reads are sampled according to a Poisson process with a background rate of mu t, and a delta IT, which is piecewise continuous, a multiplying factor that is denoting whether there's a copy number change there. And this has change points tau I, which belongs to a set of change points that are shared across cells. They're shared across cells because the cells evolve according to the same evolution. So, they tend to have breakpoints at locations that are, shared by some ancestor, right, in their evolutionary process. But if the read Yij, let's say, overlaps, I is for cell and J is for read. If it overlaps with a heterozygous site, and we know these a priori, so we assume that you sequence the blood of the patient, and then you can call the heterozygous sites, or that you have some way of getting these germline heterozygous sites to begin with. Then we let ZIG to be the indicator of whether it is the alternative allele. Right? So each read that covers a heterozygous site also gets a ZIJ indicator. And this indicator is either 1 or 0. It depends on whether the read, the I, which is whether the read falls on the major haplotype. So whether the B allele, falls on the major haplotype. And it also depends on whether the major haplotype is amplified or, what copy number it is, right? So whether it's a 1-2 ratio or 3-1 ratio or whatever. So theta is the underlying major haplotype proportion that we want to estimate, and I is a sort of a nuisance, underlying coin flip of which whether the blue or the red haplotype the B allele fell on. So our parameters of interest is theta and delta. And what is important to know here is that we are looking at single cells. Now in single cells, things are discrete. So it's not like the bulk sequencing scenario where you're looking at a mixture, so you can have continuous values. So in single cells, your delta and theta values fall on one of these canonical points, right? So if it's a diploid, so 1, 1, this gray or black circle, then your theta is 1/2, right? Because 1 out of 2 is B allele, and rho is 1 because you don't have a deletion or amplification. Now, if you do have a deletion to 1 copy, then your rho becomes 0.5 at the purple dot, and then your theta is 1. If you have a copy neutral LOH and you're at the cyan point, right? So your rho is 1 because you didn't have a change in total copy number, but your theta has gone to 1. Now, if you have an amplification, you can be at any of these points, right? 1/3, 2/3, or or if you have 2 copies of the amplification, then it could be either 1/4, 1/2, or 3/4. So at the single cell level, your underlying state is discrete. So given this model, I will just give you a quick rundown of how Alleloscope works. Okay, so we start with the input matrices, which is snipped by cell. Okay, the alternative versus the ref, the A versus the B allele. And then what we first do is we pull across cells to get the bulk, relative coverage, and the A allele frequency, or the B allele frequency. We just showed the A allele frequency here. And then we do a segmentation because there's existing methods developed by our group or others that do this segmentation in bulk. Because we reason that if there is a subclone that has a copy number change, it should register a signal at the bulk level if the subclone is not too rare. So if you are working with ATAC-seq data, then we recommend using the whole genome sequencing or whole exome sequencing that is matched with the sample to do this segmentation, so that you start with regions that are defined by DNA. And then for each region, what we do next is we simultaneously phase the SNPs in that region and infer the cell-specific major haplotype proportion by iteratively pulling across cells for the phasing and pulling across SNPs for the cell major haplotype proportion estimate. So this is, it's nice that in the model, there's a natural way to do this to the EM algorithm. Now, just to a reminder, what we mean by theta is the cell-specific major haplotype proportion. So in a normal cell, it should be 1/2. In say, an amplification is 2/3, and deletion is either 1 or 0. Once we have the data for each cell, we can now, okay, let's show what happens next. What we can do now is we can look across the cells to identify the normal cells. Now that seems like an obvious thing that should be given to you, but oftentimes you don't know the normal cells among the cells that you sequence. And you can't look at the total copy number to identify the normal cells, because in the estimation of total copy number, there's a lot of sequencing biases that just, it's actually pretty hard to estimate, because the reads you get, the background rate of reads is not constant even for the normal cells. So we use the data to identify the normal cells, and once we have them, we get the normalized coverage, which we can then just plot against data to get points on this curve. On this 2D plot. And then what we do is we cluster to get the subclones. So, I think this might become clear if I show you how it works on a real data example. Okay, so let's look at this gastric tumor example. And what you see here is what we get out of the 10x software. So 10x is a company that does single cell DNA sequencing, and they have a software called Cell Ranger that gives you this plot. And what it shows is just against genome position. So the rows are the cells and the columns are the genome positions. And then you see it like an orange, a bar if there's a copy number change there, and a blue bar if there's a deletion. Now this turns out to be, I think this is a male, so that's why there's a blue bar at the X, right? Because there's only one copy of the X chromosome. But you see that from the 10x software, you see that this is actually quite a silent genome as far as tumor genomes go, in the sense that there's actually not a lot of copy number changes, right? There's a 7 amplification, 8 amplification, 20 amplification, and 21 deletion, but not much else that is going on. And that is what we expect, because this is what's called a microsatellite unstable tumor. And in microsatellite unstable tumors, there's not a lot of copy number changes going on. So we expect that clonally, there's not very much diversity in terms of copy number. But let's see what happens when we look at this sample through the Alleloscope. Let's focus on this amplification of 7. Okay, so as expected, we see this gray cluster at rho equal to 1 and beta equal to 1/2. Now those are the normal cells. Okay, but then we see 2 additional clusters. We have expected to see this cluster, which is the dominant cluster, and that is the amplification of the major haplotype, the gray haplotype. But what we were surprised to see just right off the bat that there is this other cluster that is mirrored to it. And this is another subclone that has amplified the minor haplotype. And, so Ben, we were looking for these mirrored subclones. That's one reason why we did this analysis, but we didn't expect to see it right off the bat for the first sample we look at, literally, and for a sample that is supposed to be genomic, pretty silent. Now let's look at this chromosome 8 region. For this chromosome 8 region, we saw this amplification, right? This subclone that carries the amplification, which is expected. The gray, again, is the normal cells. But we see these very clear subclones that are centered at these other canonical points. So for this region alone, it seems as if there are 4 tumor subpopulations. Now, we would not see this diversity if we were to just look at total copy number. Maybe we would see these, but the evidence, we wouldn't be so sure of them. But when we look at the B allele frequency, we are very certain that these are subclones. Now, so I'm just showing you, what they would look like. Now, for the deletion also, you see this mirrored subclone here. So it seems as if, at least for this sample, mirrored subclones are the norm rather than the exception. And that is what we were surprised to see. Now, what about other samples? We explored a range of cancers: colon cancer, gastric cancer, breast cancer. And for each cancer, you have many regions across the genome. So I only have space to show you 2 regions per cancer. So we chose the interesting regions. And, for each region, if you were to just look at it through bulk sequencing or microarrays, right, you would just see that maybe, these are the amplifications. But if you were to look at single cell allele-specific copy number, you will see that they are very diverse. So for example, for this colon cancer, right, you have an amplification, but you also have an LOH, a loss-of-heterozygosity in this region. That's the cyan cluster. And you also have 2 copy amplification where there seems to be 2 haplotypes that are affected, and 3 copies. And then for this gastric cancer, you see an amplification of the major haplotype, but there's a mirrored subclone that has the minor haplotype amplified. And then look at this amazing diversity, right? So you see that, for example, for this cancer, this breast cancer, this one is analyzed by the Chiso paper, that this is their data. And they only said that there's there's a mirror subclone, but what we see is that there's 1, 2, 3, 4, 5 different subclones for this cancer, for this region alone, right? So this is really amazing. We were just, just really excited about this because never before has this level of subclonal diversity been teased apart by copy number analysis. So then we started asking, when you, in genomic data, when you see something that's really interesting, then you start also getting worried that it might be artifacts, right? Because it's repeated in every sample. Maybe there's something wrong with the experiment that's causing these clusters. So we asked our collaborator to do a link, what's called a bulk linked read sequencing experiment, where you sequence the cells in bulk, but you link the reads that are on the haplotype. So that's a short way of saying this experiment. So we know from this experiment what the phases of the SNPs are. So we did matched bulk linked read sequencing that allowed us to phase the SNPs. Now remember, the hard part about this analysis is the fact that the SNPs are not phased. Once we have the phasing, we can get much more certain, it's just a very direct estimation problem. So here's just a study, the sample I showed you earlier. Right, so the top is the Alleloscope estimates, and the bottom is the estimates you get using a linked read information. And the bottom plots are, this is important to note, they are colored by the same coloring as the top plots. So you can use it to see whether the clusters correspond to each other. And you see that basically with linked reads, you discover the same subclones, right? But not many labs can do this linked reads analysis. And that's why we don't want to have to rely on this information in practice. Right, but this gives us a validation that these subclonal structure that we see is biologically real. And then we did link reads for the 6198 sample and this P56645 sample, and they were all validated. So we were able to be quite confident in our paper that these are real subclones and not just technical or estimation artifacts. Okay, so, and what's important is that some of these samples that we analyzed are metastasized samples. Now, what is often believed about metastasis is that it's a bottleneck event, an evolutionary bottleneck event. And then when you get the metastasis sample, it's genetically very homogeneous. But you see that that's actually not the case here, right? So you actually have a lot of subclones with different copy number changes in the metastasized sample. So that just shows that this process of chromosome breakage, and it shapes the evolution through the entirety of the disease, not just in the primary tumor. So this allows us to take a tumor, and previously you could just see this bulk copy number profile. This is what we always look at when we look at cancers, like say 5 years ago. But now we can estimate the allele-specific copy number and assign these regions to allele-specific copy number states, and that allow us to cluster them into subclones. And then we can take these subclones and analyze the evolutionary relationship between them. So here's a tree that we constructed, where at each branch of the tree, we put down what is the actual copy number event that leads to that subclone. Okay. So that's the, sort of a rundown of what we can do now with single-cell DNA-seq data. But, I'm going to go on to the ATAC-seq. So the ATAC-seq, remember, we want to look at the epigenome and see how these two processes intertwine, right, to shape the cancer evolution, right? So remember, our model doesn't actually require you to have homogeneous background rate of reads, right? And in ATAC-seq, what you observe is peaks. So what existing data analyses pipelines do is they just take a smoothing window, a running window across the genome, and use the window mean to try to get the copy number. But that is very problematic, because even between normal cells, so here I'm showing an endothelial cell as compared to a fibroblast, even between normal cells, there are chromosome level remodeling events that basically look like copy number in ATAC-seq, right? So here, for example, you would think that these are deletions and these are amplifications if you were not careful, right? So we thought that we should add allele-specific information into this. And so we applied our method. I'm not going to look at this. This is example in our paper. I'm not going to look at this. I'm going to go back to this gastric cancer cell line that I just showed you in the previous slide. Now, remember, we already analyzed this using single-cell DNA sequencing data, and that allows us to gauge how accurate the single-cell ATAC-seq analysis can be. So we applied Alleloscope to single-cell ATAC-seq, and here are the sort of the relative coverage versus the major haplotype proportion plots. And you see that the clusters are more diffuse, and that is because for single-cell ATAC-seq, we have about 10 times less reads per cell to do this estimation. But the clusters are centered at the positions that you expect them to be given the single-cell DNA-seq. So we were able, even though for each region alone, there's too much noise to call the copy number states precisely. But when we pull data across regions, we are able to cluster the cells into subclones. And that allows us to basically integrate DNA copy number and chromatin remodeling information. For example, we can basically look across, compare two different subclones and ask, between these two subclones, what is a peak level remodeling event that is significant after accounting for the underlying copy number changes, right? Because just due to the copy number changes, you're going to get a lot of peaks, ATAC peaks that are significant differentially enriched between subclones. But that's not very meaningful because it's just reflecting the copy number change. So we are asking what are events that persist after you have controlled for copy number changes. And then so here in the Venn diagrams, you see on the left are those that were significant before controlling for copy number change. And then this is significant after, and these, on the right are the events that unique that you can detect after controlling for copy number changes. Okay, so we're right now applying this to many data sets in the human tumor atlas network, and I just want to summarize that. This allows you to basically do this analysis in low coverage and ATAC-seq data, and that allows you to sort of dissect the contributions of these two factors in intratumor heterogeneity. So in the next, I'm going to go very quickly through our single-cell RNA-seq analysis. So why is CNA detection in single-cell RNA-seq so hard? The reason why is because gene expression varies over a much larger dynamic range. And then you have to also deal with transcriptional stochasticity when you look at gene expression. Okay, so that makes total copy number estimation hard. But also, we rely on reads that cover heterozygous sites, and there's not many of these in coding regions, which is why allelic information is sparse, and that makes allele-specific copy number estimation hard. Okay, so existing method for RNA-seq also is kind of like what I showed you for ATAC-seq, where they just use smoothing windows to try to pull information across adjacent genes in estimating the copy number change. So here is an inferred CMV result. Inferred CMV is what everybody uses. And you see that here are the normal cells and here are the tumor cells. So you would think by looking at this that there's all these deletions and amplifications going on in the genome. But we actually sequence this sample using single-cell DNA-seq. And you see that there's basically only 4 regions that you have copy number change, and they are right here, right? So that means that all of these other are background noise that you sort of didn't filter out. So we developed a method based on, I'm not going to describe it because it's sort of similar to Alleloscope, but it takes advantage of unique things about single-cell RNA-seq, and we were able to basically recapitulate what we find in single-cell DNA-seq. And then we can apply this to spatial transcriptomic data. So here is an H&E image of a breast cancer. We can identify copy number changes in this tumor. And that allows us to identify the subclones, so 3 different subclones. There's more here, but we're just going to highlight these 3. And then, between the subclones, there's credible evidence that there's copy number changes, right? So here's just, you can sometimes see the density plots much easier. And then if we map these subclones back onto the spatial spots, we can now gauge where the subclones are in space, how have they expanded, and whether they are intermixed or separated, etc. Now, I want to note that we didn't actually use this spatial information in the subclone estimation. So, there's like one other method out there that's tried to sort of smooth across space, and that allows the estimates to be more robust. We don't do that because we don't want to a priori assume that the subclones are not mixed across space, right? They might be mixed. So, but it's kind of reassuring to see that without using the spatial smoothness information, our subclones do have this spatial localization. And that gives us more confidence it's real. Okay, and you see here is a pathology annotation. We had an expert pathologist sit down and highlight where the tumor regions are, and you see that these are indeed coincide with what we say is the tumor regions. Okay, so just a summary, we developed a method for single-cell allele-specific copy number estimation. It finds pervasive evidence of multi-allelic copy number aberrations and just shows that there's these highly mutable regions that are seeding new subclones. And when applied to ATAC-seq, it allows us to interrogate the contributions of chromatin instability and epigenetic plasticity in tumor evolution. And we have been looking at the RNA-seq and spatial transcriptomic data, and there is a beta software here available, and you can stay tuned and you can test it. And I want to thank you guys for, I know it's hard to always sit through a Zoom talk. I want to thank you guys for being here, and also the people who have contributed to this work, mainly Chi and Wu, who is a graduate student who did a lot of the heavy lifting, and also my collaborators at UNC and Stanford's lab, Hannah G's lab, did a lot of the validation and data generation for this. Okay, thank you.
Thank you so much, Nancy. This was wonderful, wonderful talks. A lot of great stuffs are ongoing. So any question from audience? I guess if you want like to stop sharing, or if you want to refer to it, we can refer back to it as well.
Yeah.
Let me start by asking a question, and audience could also like unmute and ask. So you're looking at this eventually the subclones, and in these like subclones that you show, you're saying that maybe these cells at this loci is having this copy number variation differences. Is there any way that we could connect all of these across the entire genome and say like this one cell or these 5 cells are the ones that belong to a new clone, and this new clone has all of this feature? Or is it like at the region level, or we can only look at it at the region level?
Yeah, so you can connect this across regions. I mean, because for each cell, you basically have a vector of what copy number state it is across regions. You can use this to sort of cluster the cells or perform phylogenetic analysis on the cells to try to get at, what are the subclones and how the subclones are related to evolution.
And then, right, and then in that sense, are these subclones kind of clear where now they are coming from the main clone and evolved from the main clone?
Yeah, so right, we try to do that in our paper. We try to, because when you have these subclones and you always have to figure out what, reconstruct the evolutionary history and try, what are the events that led to these subclones. You're never sure, but, people do this in phylogenetic analysis all the time, right? And when we connect, look at animals and, you have to figure out what happened in the history. So there they use they have a lot of assumptions, like, for example, the infinite size assumption, that, constant molecular clock assumption. None of these hold in tumors, right? In tumors, because of the high mutation rate, and you see that these copy number changes always affect the same region. So it's not like a one-hit hypothesis. So we, in our paper, put down a few evolutionary rules that we thought make sense for copy number change, which some of them are very obvious. Like if a haplotype is deleted in a clone, you cannot get it back in its descendants. That's, so you cannot go from a 0 to a 1. So this is one rule that, and then there's another rule where, if you go from 2 copies to 4 copies, and there's an intermediate clone with 3 copies, it should go to 3 copies first, unless there's a genome doubling event, in which case then this doesn't apply. So there's these rules that we wrote down for copy number evolution that sort of help us get at the phylogeny. But this is also a program that's very hard.
This is very, very exciting. So any question from audience? I know that, don't be shy, guys. If you have any question, feel free to unmute and ask. So while I am waiting, I'm going to ask one more question. So for these mirrored subclones, that's a very interesting concept. Do you know that whether any of them were associated now with functional change, or have been in the area that there has been any functional association to them?
Yeah, so we don't know. The short answer is we don't know. And the reason why we don't know is before these mirrored subclones were sort of this mysterious event that that a few studies have sort of found they were not broadly implicated because they were underdetected, because you don't see them unless you have both, single cell and allele-specific copy number, or if you do multi-region sequencing, so you slice a tumor up and sequence multiple regions. It's not until recently when studies doing this have been published. So there's been a few sightings, but it's like, it's like the Bigfoot. You see a few, you don't actually know what they are. So what we hope is that now that we have shown that these things are sort of the norm, and when you have, among the cancers we looked at, when there's an amplification or deletion, it usually happens multiple times and is seeding subclones with mirrored events. Now we can actually, I think, ask this question, which, that you ask, which is, I think, a very, very pertinent question in cancer evolution.
Very, very exciting. I'm going to ask one more question. I think that unless anyone else wants to ask question. One more thing is, like, have you had a chance to take a sample from metastasized site and the primary size and see whether any of these like rare subclones are now expanded in the metastatic site?
That's such a good question. And, that's exactly what these studies should do and what we want to do. There has been quite a number of studies, not from our group, that has done such things, but using different profiling techniques. Like they may do targeted sequencing of oncogenes and then look at point mutation of these oncogenes, and they have multiple time points and thus are able to look at the sort of clonal expansion across time. You can imagine that such studies are very hard because it's not always the case that you can get 2 samples per patient, right? So you have to follow up the patient within the same medical center, right? Sometimes they shift to different places, and so you have to do longitudinal follow-up for cancers, and so basically that explains why the first such studies come from studies of leukemia, where you use just a blood draw, which is much easier, right? Instead of like an operation to get the cancer out. So others have done this, and they have shown that it's a very informative analysis to do, because you can just look at within a cancer a clone that has expanded, and that tells you, and so why is it that clone? And then that tells you what is actually happening in the patient. There's this Human Tumor Atlas network, which is this broad network that across institutions where people collect data. And one of the priorities is to look for these kind of longitudinal multi-time point samples.
Right, either that or the drug resistance, those clones that are becoming the drug resistant. Wonderful, wonderful.
Right.
Thank you so much.
Thank you. Yeah.
Any last minute question before we adjourn? Okay, I don't see anyone.
I found when I run these Zoom seminars, I just find that the shy people are more shy when it's on Zoom. I guess when we hear ourselves and see ourselves talking, it's always harder.
So I think that this has been extremely informative for a lot of folks in our department. So thank you so much. Let's thank our speaker one more time, and hopefully next time we'll have you in person here.
Thank you, Majid. All right, see ya.
See you later. Bye.
Bye.